Okay, so in this question here, so again, I've been asked by a student to show you the question with regards to neural communication. So let's have a look at this then. So you're told in the question that you have got the greater blue-ringed octopus. So they've given you some information. So straight away, I would be adding a little bit here that says, well, they've given us our genus and our species name for this octopus. They may ask me a question later on about that. The other thing that they could do is they could give me some information, as they have done here, that tells me that this octopus is a venomous animal. Now, its bite contains this tetrodotoxin. Now, most students look at that and go, well, now I don't understand what this question's about because my teacher hasn't taught it to me. Well, what you have to understand is that this tetrodotoxin is, as you've been told, a neurotoxin. They've already told you it's to do with your nervous system. So a lot of the times they will give you lots of information in the question, and what they're actually wanting you to do is apply your knowledge to the situation. So if we then look at this neurotoxin and what does it actually do? So they've told us in the question that they discovered that the effects of the neurotoxin on the nerve cells, again, little hint here, is that it will bind to external surface of voltage-gated sodium ion channels. And that's on the axon membrane. That binding is going to change the tertiary structure, and this means that the channel cannot open. So they're giving us lots of information in this question and then asking us to use that information and our own knowledge to explain how that's going to affect the activity of a neuron. So let's just go back and look at our neurons and remind ourselves about the structure of a neuron. So you should know that the structure of a neuron is that you have got your axon is inside and you have your outside, which is your external environment, so extracellular fluid. So you can see on my diagram that I've got my inside of the axon and my outside of the axon. Now, you should remember, if you looked at my previous video, that there is the resting potential, where the potential difference across the membrane is minus 70 millivolts. And that is because you have your sodium-potassium pump, and there's 3 sodium going to be pumped out, 2 potassium going to be pumped in. Therefore, maintaining a concentration gradient of sodium being high on the outside and low on the inside, and potassium being high on the inside and low on the outside. And you should remember that your potassium, if the channel is there and it is open, it will allow for the facilitated diffusion through that carrier protein or channel protein to allow the potassium to go through. So we therefore know that our resting state is to keep a potential difference between our membrane, more positive on the outside, more negative on the inside, at about minus 70 millivolts. And the fact that we want this electrical chemical gradient, we want this concentration gradient of ions to be different. Now, they're expecting you to remember that you have your sodium-potassium pump. You also have these voltage-gated sodium channels and voltage-gated potassium channels. So because we have these channels, and these channels are voltage-gated, that therefore means that they're only going to open or close when we get to the right potential difference. So if we look here, then we can see if I depolarize the membrane take it away from its resting state, but I don't reach that threshold value, remember, then that will be brought back down to the resting state by my sodium-potassium pump. If I, again, depolarize the membrane, but I'm depolarizing it not quite enough, it will be brought back down to the resting state. So if the stimulus is not going to reach that threshold value, those voltage-gated channels are going to remain closed, and the membrane is always going to be brought back to that resting state. So if I do happen to reach that threshold value, if the depolarization of the membrane reaches that threshold value, then that is going to trigger that action potential. Now, an action potential starts with depolarization. So these voltage-gated sodium channels are going to open. The sodium is going to flood into the axon down that electrical chemical gradient. So if we go back to this question then, we're being told that this chemical is going to bind to these voltage-gated sodium ion channels. It is going to change their tertiary structure. So if it's going to change their tertiary structure, it's therefore going to affect their ability to be able to let sodium move. So to answer this question then, we need to think about that. If we're changing the tertiary structure, that is going to, as it already told you, stop the channel from opening. So if there is a channel, my voltage-gated sodium channel cannot open. If that voltage-gated sodium channel cannot open, that therefore means that sodium is not going to enter the cell. So voltage-gated sodium channel cannot open, therefore no sodium will enter the cell. Because remember, our resting state was that we would have a high concentration of sodium on the outside, a low concentration of sodium on the inside. And if we have our voltage-gated channels and those voltage-gated channels open, the sodium can move in to the axon, and that will then depolarize the membrane. Well, we've been told here that those voltage-gated sodium channels cannot open. So if they cannot open, no sodium can enter the axon. Well, if we then go back to looking at what happens if no sodium enters, well, if no sodium enters, we're not going to reach that threshold value. So because we're not going to reach that threshold value, that therefore means that so in this case, we do not reach. So if there's no sodium that's coming in, we cannot reach that threshold value. We have not depolarized the membrane enough so that we've reached the threshold value. In that case, we cannot trigger the action potential. If we can't trigger the action potential, we don't have the depolarization. And if we look at this then, when we open those voltage-gated sodium channels, the sodium comes in. Now, what that sodium will then do is, because it's flooding into the axon, we're getting a high concentration inside the axon. It will then increase the potential difference inside the axon. Well, what that then does is that gets to the peak, which is in this case on my graph, a plus 40 millivolts. That will then cause those voltage-gated sodium channels to now close. That results in repolarization. Our voltage-gated potassium channels open. We are now having the potassium ions flood out of the axon, down their electrical chemical gradient, and then they will diffuse out, and they will therefore lower that potential difference back towards that minus 70 millivolts. But we overshot it. So what we have to remember is that when we do an action potential always has three parts to it: depolarization, repolarization, and then we get to this hyperpolarization, where we have overshot that minus 70 millivolts, and we have hyperpolarized the membrane. So in this case, our voltage-gated sodium channels remain closed. Our voltage-gated potassium channels are also going to close. And then our sodium-potassium pump is going to bring that back up to the resting state. Now, the reason this has to happen is because we know that when the sodium comes in, the sodium is going to move and diffuse to an area with a lower concentration. That will therefore depolarize this section of the membrane. And if it reaches the sodium channel, the voltage-gated sodium channel, if that reaches the potential difference of the threshold value, that will then cause that membrane to open. And therefore, this is no longer at that resting state. So the resting membrane becomes depolarized towards that threshold value by this local current, and that is what we know as positive feedback. That will then cause those channels to open and the sodium will come in. But the sodium, when it comes in, that's going to diffuse from a high concentration to a low concentration, and again, from a high concentration to a low, and you will have seen that going in both directions. So the reason we've got that hyperpolarization, the refractory period, where these voltage-gated sodium channels remain closed, is to ensure that the action potential is going to be propagated in one direction. So if we go back to the question then, and we actually look at what we're being told in this question, we know that our voltage-gated sodium channels are going to remain closed. Therefore, no sodium is going to enter. Therefore, we're going to have no depolarization of the membrane. That's going to mean that we are having no action potential, because it will not reach the threshold value. And therefore, if we're not going to get an action potential, that impulse is not going to make its way along the axon from the cell body to the end of that axon terminals. So therefore, what we have to understand is, if we're not propagating along the membrane, because what we would do is an all or nothing response. We depolarize to the threshold value, and then it's an action potential. So the action potential, depolarization, repolarization, hyperpolarization, back to the resting state, is an all or nothing response. It is a propagation all the way down through the axon. So if we've got a myelinated neuron, that's where we've got up to 20 layers of the membrane, those phospholipid bilayers wrapped around that axon membrane. That therefore means that the only places where an action potential can occur, and only place where the depolarization of the membrane occurs, is at the nodes of Ranvier. So the nodes of Ranvier is the point where we're going to see those voltage-gated sodium and potassium channels. So we have the depolarization only occurring at the nodes of Ranvier, and therefore the action potential would jump from one node to another, which is known as the saltatory conduction. But we have that in our membrane. We have not propagated along. So we had our axon coming down. We'll get to that synapse, and then we have our postsynaptic membrane, which will then lead on to our next neuron. So what we're saying is, if we have an action potential make its way down, propagate down through the neuron, what that will then do is that will then cause vesicles containing neurotransmitters to fuse with the membrane. And if they fuse with the membrane, that will result in the release of the neurotransmitter into the synapse, into the synaptic cleft, and that can then bind to receptors which are on the postsynaptic membrane. So if we've got receptors on our postsynaptic membrane and we release the neurotransmitter, if that neurotransmitter can diffuse across and bind to that receptor, that could cause that channel to change its shape and open. And if that changes its shape and opens, that therefore allows for the sodium to come in. The sodium comes in, an action potential can then be generated and triggered to make its way down that postsynaptic neuron. But remember, going back to the question that we had before, if we are going to prevent the voltage-gated sodium channels from opening, we're therefore not going to allow sodium to enter. There's going to be no depolarizing of the membrane, no action potentials because we've not reached the threshold value. Therefore, this will not happen. So there will be no releasing of a neurotransmitter. And if there's no releasing of the neurotransmitter, that will therefore result in no threshold value being reached in the postsynaptic membrane. And therefore, if there's no threshold value in our postsynaptic membrane, that therefore means there's no impulses being passed on. And just to make sure I do this properly, make sure whenever you do a label that you get your label line to only touch the area that you're looking at. So that is how to answer that first part of that question. So now if we look at the next question then, so the next question says, a common cause of death from TTX poisoning is suffocation, not enough oxygen as a result of paralysis of the diaphragm. So in this case, because it's paralysis of the diaphragm, what they're actually checking is, do you know anything about your diaphragm? So the diaphragm, you should know, is part of your exchange surfaces topic area. And therefore, what is the diaphragm? Well, we know that the diaphragm is going to be contracting to increase our volume when it drops down. So when the diaphragm contracts, what that then does is that increases the volume inside our lungs. And what it then does is that then affects the pressure. So if we are increasing the volume, we're going to decrease the pressure inside the pleural cavity. We're going to decrease the pressure inside the lungs. That's going to cause air to flood into our lungs. So the air is going to flood in. That will therefore allow for gas exchange because of our concentration gradient. Because when we get down to our alveoli, remember we want oxygen to come in so that it can diffuse into our bloodstream and diffuse across into our erythrocytes, and we want our carbon dioxide to diffuse out. So the only way we can ensure that that happens is if we actually have a concentration gradient. Well, going back to the question, we've been told that our TT poisoning is actually going to paralyze the diaphragm. So how is that going to paralyzing the diaphragm going to cause the person to be suffocated? Well, we've got no change to the volume in the thorax. We've got no change to the pressure. Therefore, we've got no air moving in. No air moving in, no concentration gradient. That is therefore going to cause someone to suffocate. So again, this question, yes, the previous part was about neuronal communication, but this question was actually testing your knowledge on exchange surfaces. And do you know how they link together? Well, if I'm going to do exercise, what I need to understand is that for me to do exercise, my muscles will be contracting. Because they're going to be contracting, they need more ATP. Therefore, more respiration needs to occur. Therefore, they need a supply of oxygen and glucose for respiration to occur. Well, because I'm doing more respiration, I'm going to have more carbon dioxide. That is toxic. I need to remove that from the body. Well, the key thing to remember is my body is going to be detecting changes in my hydrogen ion concentration, and therefore detecting those hydrogen carbonate ions because of dissociation, and that's going to result in my body responding by my neurons sending impulses to my diaphragm, meaning that I will contract the diaphragm at a faster rate. Therefore, I can end up with a breathing rate that has increased, and also the depth at which I take the volume in changes. So it's important that you note there's an interlink between animal responses as well as your exchange surface topic area. Okay, so moving on to the next question then, this one here, it's now asking about the reducing the speed of conduction in the Purkinje fibers. So it's asking you to suggest and explain what is going to happen to the heart rate. So reducing the speed of conduction. So for this one, it's expecting you to understand, let me just get up my notes for you on this one, that you have your sinoatrial node, your atrioventricular node, and your bundle of His, your Purkinje fibers, and your conducting fibers. So there's an expectation that you understand. I'll just add this in to the part here. There we go. So if I just bring this in here, if we look then, you should know that you have your sinoatrial node. That is going to send electrical impulses across the atria. You also should know about your atrioventricular node. That is going to respond by that impulse that's come to the end of the sinoatrial node, then being passed on to the atrioventricular node, then goes down the bundle of His, down the Purkinje fibers, and there is a non-conducting tissue in between your sinoatrial node and your atrioventricular node, therefore delaying the impulse from going from the atria, the contraction of the atria and the ventricle, because you want a slight delay. Well, this question specifically tells you that you're already at the Purkinje fibers. Now, a lot of students tend to trip up because they go, oh, I'm just going to tell you everything I know about the sinoatrial node, the atrioventricular node, and so forth. But what they've already mentioned in this question, in the stem of the question that you have to read, is that the TTX is known to reduce the speed of conduction in the Purkinje fibers. So therefore, where are we actually slowing the impulses? We're slowing the impulses down to the ventricles. We're slowing the impulse from our atrioventricular node to the ventricles. So you're first thing to consider when answering this question is the fact that if we are slowing the impulse from our atrioventricular node to the ventricle, because that is where the Purkinje fibers are, that is going to result in a slower oh my word, this is where my dyslexia kicks in massively. Slower ventricular contraction. So I've now got a slower ventricular contraction. Therefore, that is going to mean that my heart rate will be slower. If I've got a delay between the ventricle contracting and then the ventricle contracting later on, that is going to result in that slower heart rate. So I hope that has been helpful.